right, we're back for another lecture on nucleic acids. And the specific nucleic acid we're still talking about is DNA or deoxyribonucleic acid. And today we're going to talk about how DNA replication or how DNA makes a copy of itself. So basically, we're going to look at the process of how one strand of DNA, after DNA replication, you end up with two identical copies of that original strand. So DNA has to copy itself every now and then. So this is the process of DNA making a copy of itself. All right, first, just a little review. See if you remember. What is the backbone or the sides of DNA made out of? So you see this yellow circle, this little green, like kind of house looking structure. You can see the same thing over here. The backbone. What are the sides made out of? Remember, the little circle is a phosphate. So I'll put a P for phosphate. And remember this little greenhouse? That's deoxyribose sugar. So the sides of DNA are made of phosphate sugar, and same thing over here, phosphates and deoxyribose sugar. Remember, inside here, those are the nitrogen bases. Do you remember what base goes with A or adenine? If you said T or thymine, you'd be correct. If there's a cytosine on the left, C always goes with G. There's going to be a guanine on the right. C on the left again, G on the right, because C goes with G. T on the left, there's going to be an A on the right, because thymine and adenine go together. G on the left, there's going to be a C on the right. All right, so there's the structure of DNA again, phosphate sugar on the sides, nitrogen bases in the middle. All right, so what is DNA replication? It's a process where DNA is going to make an identical copy of itself. The cell is going to make an identical copy of its DNA. So remember, you're going to start with one strand of DNA. Remember, it's a double helix. It's got two sides. And at the end of the process, you're going to get two exact copies. So one strand of DNA is going to become two strands of DNA. It happens before cell division, which is mitosis and meiosis. We'll learn that next semester. Why is it essential? Growth. When an organism gets bigger from a baby to an adult, you need more cells. Every time you need more cells, you need more DNA. Repair. Your skin cells only last about 2 weeks. So if you didn't make new skin cells after about 2 weeks, you'd have no skin. You got to repair. You got to replace those dead skin cells, and also reproduction of the organism. So if you have dogs and you want to have puppies, you got to make new cells, and that's what's going to be called meiosis, and you need DNA replication for that. All right, so we're going to go over the process of DNA replication. All right, so before I get into how DNA actually makes a copy of itself or replicates itself, you need to know a term called semi-conservative. So DNA replication is semi-conservative, and it's really simple. All right, so here's the original strand of DNA. DNA is double-sided. It's like if it's an A over here, it's going to be a T over here. Remember, it's got the sugar phosphate backbones on both sides. All right, so semi-conservative. First of all, there's an enzyme. I'm going to kind of draw them in right here. It's called helicase. Its job is the double-sided DNA. It goes right down the middle and unzips the DNA. So you can see right here, they used to be together, bonded together. Well, helicase is moving up and it's unzipping it. So instead of a double-sided strand of DNA, now we have two single-sided strands. So now when we're making new DNA, you can see that the two new strands down here. The red part is brand new. It's newly synthesized strand. It's brand new DNA. But the other side, the purple side, that's not new. That came from the original strand of DNA. So all semi-conservative means is when you make two new strands of DNA, half of the strand is going to be brand new, it was just made, where the other side of the DNA strand is old or conserved from the original strand. All right, so now the steps on how DNA actually replicates or makes a copy of itself. Step one, DNA gets unwound and unzipped. And you can see the enzyme right here, helicase. So here's DNA. It's double-sided, remember? A goes with T, C goes with G, and they're bound together with what's called hydrogen bonds, weak hydrogen bonds. And DNA's job is it's right now it's moving this way and it's going right down the middle and unzipping the DNA, creating two single strands of DNA right here. And then another enzyme is going to come along and add the other half, as you can see, is being added here and here. All right, so step one, the DNA, just like unzipping your sweatshirt. This little thing you hold on to, that would be helicase. It goes right down the middle and unzips the DNA, creating two single strands. So that's step one in DNA replication. All right, step two, elongation of the leading strand. So you have a leading strand and a lagging strand. Regular bio, don't know if you have to know this, honors you will. All right, so right here, remember here's helicase right here. It's moving this way from right to left. And so you see here the DNA is still double-sided, or helicase is going up and just unzipping it, creating two single-sided DNA strands. You can see over here, single-stranded, and over here, single-stranded. Then there's this so that's an enzyme, helicase. It's an enzyme, it unzips the DNA. Then a second enzyme called DNA polymerase. You're only going to need to know two enzymes, helicase and now polymerase. Polymerase is going to come along, grab these free floating nucleotides. Remember, nucleotide has a phosphate, deoxyribose sugar, and one of four nitrogen bases. So right here, it's grabbing a yellow thymine and it's attaching it to a red adenine. So it's going to grab these free floating nucleotides and expose them, or bond them with these exposed bases. So basically, it's making the other half of DNA. So you can see it's single sided here. Once polymerase comes through, it adds the other half of the DNA strand, making it double sided again. Now, the reason this is the leading strand up top is because polymerase is traveling in the same direction as helicase. So as helicase unzips the DNA, polymerases can continuously follow them and keep adding the other half of the DNA. It doesn't have to stop. Now on the lagging strand down here at the bottom, I'm going to show you why that's a little different. So DNA polymerase, all it does, helicase unzips it, polymerase comes and adds the other side for each of the strands of DNA. All right, step three, elongation of the lagging strand. So remember the leading strand was up top. Since polymerase is moving from right to left on our screen the same way that helicase is working, as helicase unzips, it can just keep following them and add the other half, the other side of the DNA. It does not have to stop because as it unzips, it just keeps following them and keeps continuously adding the other side. Now on the bottom, this is called the lagging strand. Polymerase this time can only work in the opposite direction. So helicase is going that way on our screen and polymerase is going the opposite way. So he cannot continuously make the other side of the strand. It's got to make it in chunks. So it allows helicase to unzip, and then it's going to make a segment of DNA on this side. It's going to add the other side, not the greatest drawing in the world. And then it's gonna wait for helicase to unzip some more. And it's gonna make another segment of DNA, another chunk of DNA. So on the lagging strand, since they're moving in opposite directions, helicase going this way, polymerase going this way, it's got to allow helicase to unzip, then it'll add a little segment. And then when it unzips some more, it'll add another little segment. And those little segments or fragments of DNA are called Okazaki fragments. Now, those only happen on the lagging strand. On the leading strand, there are no little chunks of DNA because it just all gets put in in one swoop all together. But on the lagging strand, it's got to be constructed in little segments called Okazaki fragments. All right, step four of DNA replication, joining the Okazaki fragments. All right, so here you can see the enzyme helicase. It's moving this way, left to right. On the top, you have the leading strand because DNA polymerase is traveling the same direction. So as helicase unzips the DNA molecule, making it single-stranded, polymerase can just follow along and continuously add the other side of the DNA. So remember, so it starts out double-sided, helicase unzips it, and then polymerase is going to add the other half. Now that's the leading strand. Since they're both going the same direction, it doesn't have to stop. But on the bottom, that's going to be the lagging strand. It allows helicase to unzip it, and it's going to lay down the DNA in segments or chunks called Okazaki fragments. Since it's going the opposite way, on the bottom, polymerase has to work the other way. So it's got to lay them down in chunks. Now, once it lays down those chunks, the final process, another enzyme called DNA ligase, is going to come and it's going to bond those chunks together. It's going to bond the Okazaki fragments together. So now it's just one continuous strand of DNA. It's not little chunks. All right, I forgot about step five. Step five of DNA replication, proofreading and repair. All right, so here's DNA polymerase, the enzyme. Here it is right here. So it's the guy that, remember, helicase unzips the double-stranded, it unzips it, makes it single-stranded, and then polymerase is going to come and add the other half, But one thing it also does, it can also proofread. It's kind of like a spell checker. So you can see right here, cytosine is yellow. So here it messed up. It bonded a cytosine with another cytosine, C and a C. No, C goes with G. Remember, cytosine goes with guanine. So the polymerase can actually see that, and then you can see right here, boom, it made the correction. Cytosine in yellow and bonded it with not another cytosine, but bonded it with a guanine. So that's the final step. DNA polymerase. Not only does it add the other half of the DNA, it can also check for errors, and if it finds an error, it can also fix them. All right, so now we got the final product. So we started with one strand of DNA, double-sided. Remember the enzyme helicase. It's what's going to go right down the middle, breaking the bonds, and it's going to unzip the DNA, just like unzipping your sweater. It goes right down the middle and unzips it. From double-sided, it's going to make it single-sided or single-stranded. Remember, it's DNA polymerase. It's going to come and grab these free-floating nucleotides, and it's going to add the other half. So for this C, it's going to grab a nucleotide with a guanine. With the A, it's going to grab a nucleotide with a thymine. And with a G, it's going to put a C, and so forth. So polymerase is going to add the other half to both sides. And at the very end, we're going to end up with two molecules of DNA that are exact copies as the original. So one becomes two. And remember, it's called semi-conservative because half of the new molecules, the parts in purple, that's old. We didn't just make that. That came from the original DNA. It's the red in the middle that was just made by DNA polymerase. So it's semi-conservative because half of the new molecules are old or conserved from the original DNA. All right, last slide, just a review of some key terms. All right, so helicase, that's the enzyme right here. It goes right down the middle of the DNA, breaks the bonds, and unzips it. From double-sided, makes two single-sided DNA strands. Primase, we didn't talk about primase, so we're not going to worry about that. DNA polymerase, that's the guy, once it gets unzipped by helicase, it's going to come along and add the other side of the DNA. So it's going to bond T's with the A's, C's with the G's, G's with the C's, and so forth. On the leading strand, it's traveling the same direction that helicase is, so it can just follow along and just keep adding nucleotides in one swoop. But on the other side, since polymerase has to work the opposite direction, it can just lay down new DNA in small fragments or chunks. And remember, those fragments are called Okazaki fragments. And the last thing, you need to bond those fragments together. You got to make it so they're not spaces in between them. And that's where DNA ligase comes in to bond those Okazaki fragments together to make it one continuous strand of DNA. And the last part is semi-conservative. Remember, all that means is the new DNA molecules we're making, half of it is going to be old. Half of it is conserved from the original strand, and then the other half is going to be newly constructed by DNA polymerase. So half of the new strands are old from the original strand, and half of the new strands are brand new, just made by DNA polymerase. All right, hopefully you guys took some notes because there's going to be a quiz coming up right now.